Cool. Today I'm going to talk about two preprints I put out recently, and the general topic is pre-training and protein sequence and structure for protein engineering. And thanks, Nicola Zanichelli, for organizing all this, and Sergey for the Zoom link after my technical issues. So proteins are biology's actuators. Each of our cells contain between 1 and 3 billion proteins each. They're responsible for small things like structure, metabolism, and signaling. Some examples are immunoglobulin, which are the backbones of our antibodies, which are part of our adaptive immune system. Hemoglobin is one of the most studied proteins out there. It carries oxygen in your blood. Insulin is a small signaler that regulates your blood sugar levels. And these three are shown to scale with each other. So you can see some of them are much bigger. One that's been of recent interest is the coronavirus spike protein, which allows the coronavirus to enter your cell. My favorite one is luciferase because it makes firefly butts glow. And all of this diversity in function arises from 20 basic building blocks called amino acids. They're called amino acids because on the left side here, you can see there's an amine group. On the right side, there's a carboxylic acid group, and together that's an amino acid. And what makes them different from each other is the identity of the side chain here. So in organic chemistry, people use R to represent arbitrary side chains that might change. So some of these might include, for example, in glycine, a side chain is just a hydrogen. In alanine, it's a methyl group. In serine, it's a carbon and a OH. In threonine, we add a methyl to that. For cysteine, we have a SH group. And you can see there's a diversity of chemical properties and physical properties across these side chains. I don't know why aspartic acid didn't get it's highlighted. I missed one. And just for convention, each of these amino acids has a name like glycine or alanine. It also has a 3-letter code and a 1-letter code, which you use for notational convenience. And why do we want to design proteins? So we want to design proteins because maybe we want to do new chemistry. For example, Francis Arnold's lab a few years back engineered a protein that can make carbon-silicon bonds. We might want to understand function. So this is AAV, which is a virus that is of interest for gene therapy. And you want to understand how it gets into the cells it gets into and maybe target it to the cell populations you're interested in modifying. Sometimes you use proteins directly as therapeutics. This is asparaginase, which is an enzyme that breaks down the amino acid asparagine. It turns out that in many tumors, the cancer cells can't make their own asparagine, but healthy cells can. So if you break down all of the asparagine in the tumor microenvironment, it kills the cancer cells faster than it kills healthy cells. And we also use proteins as molecular tools. This is GFP, or green fluorescent protein. You can bind it to other proteins or antibodies of interest, and then you hit it with the right wavelength of light, and you can take pictures of it glowing to see where things are in the cell. And when I say protein design or protein engineering, what I mean is that in nature, the information flows from the amino acid sequence, which is also called the primary structure. Here I'm representing the amino acids with those 1-letter codes, which determines the 3D structure of the protein in most cases. And people still work on figuring out exactly how this determination is made. With AlphaFold, we're getting pretty good. And OpenFold nowadays, from this past week, we're getting pretty good at predicting the 3D fold, or at least a 3D fold from sequence or sequences. And then that structure determines the function we care about. But for protein engineering, what we want to do is we specify the function, and we want to output the sequence that will do the thing we want. It turns out that this is hard. It's so hard that if you sidestep the problem, they give you a Nobel Prize. And one way people have found some success doing this is called directed evolution. And here, instead of trying to understand how sequence determines structure, which determines function, you just kind of take a little random walk in the local neighborhood and hope you find something good. So you take a parent that kind of does the thing you want, represented on the fitness landscape on the right as being a point that's not completely off the hill, but on the very low part of the hill. And you randomly change it, just like taking little steps on this fitness landscape, and you keep the variant or the mutant that has the best result, and you repeat until you're happy with your fitness. And this works really well if you can find a parent with measurable activity, and if you have a high throughput screen, which means you can do at least 100 or so measurements a week. And in my PhD, I spent a lot of time thinking about how can we use machine learning to enable this kind of optimization more efficiently with fewer measurements. And the key here is that instead of throwing away all those measurements you made that don't improve and only keep in the best one, you take all of that information and you take those sequence function pairs and you train a model, a machine learning model that maps from sequence to function. And then you use that model to make decisions about what variants to measure next. And I'll tell a quick story about how you might use this. So this project I did a while back with Claire Bedbrook and Austin Rice and Francis Arnold and other people. So channelrhodopsins are these light-gated ion channels. They have 7 transmembrane helices. So here I'm representing the protein structure in blue and that membrane light gray. And inside that golden molecule is called all-trans retinol. And normally it's in this all-trans configuration and the channelrhodopsin is closed. When the retinol is activated with the right wavelength of light, it shifts conformations, causing the channel to open and allowing the native function to allow sodium ions into the cell. If you express these in neurons, now you have genetically targeted expression and light-activated action potentials in your neurons. So we call those optogenetics, opto for light, genetic, because it's genetically encoded. So if you want to engineer these channelrhodopsins as optogenetic tools, some goals we might have include heterologous membrane localization, meaning we want these to express and localize not in their native algae, but in mammalian neurons. Here we have an assay where channelrhodopsin that's correctly localized in the cell membrane gets tagged with green fluorescent protein, GFP, and glows green. So anywhere you see green, you can see that the cell membrane is very well highlighted here with correctly localized channelrhodopsin. You might want increased sensitivity, so you can use less light or different wavelengths of light or different on-off kinetics, how fast or slowly it turns on or off in response to light. Unfortunately, for a lot of these properties, the screen involves expressing your channelrhodopsin in cells, poking it with an electrode, and shining light on it and measuring the current across the membrane. This is called patch clamp electrophysiology. And doing this by hand, you can generally measure about 2 variants a day. So to engineer this, the slide I showed earlier said you had to start with 1 parent. We're going to start from 3 parents because 3 is better than 1. They're called C1, C2, which had a crystal structure, a sheriff, which had a very blue shifted activation and very strong photocurrents, and CS crimson R, which is red shifted. What I mean by blue or red shifted, if we look at this plot, on the X we have the wavelength of light. On the Y we have the current normalized to the current at the strongest wavelength. You can see that CS crimson R has its peak around 560, whereas the other two, which are representative of most heterodopsins, has a peak around 490. Here is some max filler current unnormalized, well, normalized by the activation, the strength of the light used to activate it. We're looking at max filler current around 1 nanoamp for all three of these parents. And these were characterized by other people not that long ago. We use a process called recombination to generate diversity. What that means is we line our proteins up and chop them up in silico so that we have this contiguous library and this non-contiguous library. Contiguous just means every block has to be contiguous in the primary structure. The non-contiguous relaxes that constraint. And this gives us 118,000 possible sequences. And if we can measure all of those, then we wouldn't do machine learning or directed evolution. We just do that, but we can't. So we're gonna try to choose a few to measure, including those three parents. We measure all the single block swaps into a parent, and then also a training set of multi-block swaps. So you can see that when you swap out a single block, most of the time you still retain filler currents, and some of them are better than the parents, but a lot of those are dead. Once you start randomly taking these much more aggressive mutations or changes, almost everything is dead, although you might get really lucky and come up with one that's really good. So based on this training set, we also noticed that physically localization is a prerequisite to function. So we make 30 sequences predicted to localize and function. Of these, 22 have filler currents, which is an improvement. One of those is the best we've seen. And all of them localize, actually. So then we take all of our training data, retrain the models, and we have these predicted probabilities of localizing versus predicted probability of having a filler current. And there's not that many in that upper right-hand corner. So the blue line, which is P1 times P2, is above 0.5. There's only 135 chimeras. So picking from that upper right-hand corner, we choose 30 more sequences predicted to have really strong currents or really fast or slow off kinetics or extremely rudder shifted, as well as two negative controls that are single blocks away from a really good training point, but are predicted by the model to be very bad. You can see here the negative controls are indeed very bad. Everything else is pretty good. And we have a bunch that are over 3 nanoamps. And these new variants are actually powerful enough to enable control of mouse neurons or mouse behavior without having to remove the skull. So you can mount the fiber optic cable directly outside of the mouse skull. And still, when the light shines, the mouse turns to the left. So this is a quick story about the success of machine learning guided directed evolution. But there's still some successes here. It turns out that for many protein properties, you can build pretty good models with fewer than 200 measurements, good enough at least to find improved things. So you can use those models to find rare improved sequences. But you still have to have starting points with activity. And we collect mountains of protein data worldwide. And this kind of paradigm so far has only used the ones we collect ourselves, about 200 points. So let's see if we can use some of that other data to do things. So we have access to these large protein databases, such as UniProt, which if you duplicate is UniProt 400, about 236 million sequences. These numbers are a little bit out of date because I made this slide like a year ago. You can cluster that or cluster it more. If you are picky and you only want experimentally verified sequences, you can look at SwissProt. So you can see most sequences we know of come from just sequencing, but we haven't actually observe that specific protein sequence. We're just looking for open reading frames in genomes. And we have structures for a subset of these deposited in the PDB. And remember, when we did our channel adoption study, we were working with about 200 labeled points. So what can we do with these databases? Well, one thing that people have been doing for a while is pretending that the amino acid sequence is a language. So we treat those amino acids like tokens, and then we use tools originally developed for language. Sorry. For example, back in 2017, 2018, several groups, including me, applied word2vec to protein sequences. So the idea behind word2vec is that similar words appear in similar contexts. So applied to protein sequences, the idea is that similar k-mers or subsets of subsequences of amino acids appear in similar contexts. So you predict the central k-mer from its surroundings, or you predict the surrounding ones from the center one. And you want to learn a vector representation that allows you to do that accurately, and then you can use that representation or embedding in downstream tasks. Then people discovered this whole deep learning thing. And another way we can do this is an autoregressive language model parameterized in neural network. So this means take your protein sequence, and given the previous token, you always want to predict the next token. And you can use that penultimate layer, once again, as a representation or embedding, or you can take your whole model and fine tune it. Then we have the neural mask language model, which really started with the BERT model, where you take your tokens, mask some of them, cover them up with a special mask token, or randomly mutate them, run them through a neural network, and try to recover the original sequence. Once again, you can use that penultimate layer output as a representation or embedding. And most of the time, people do this with what are called transformer models, which were first used in natural language processing, introduced originally as a sequence-to-sequence model for machine translation. And then people just took that encoder, that bidirectional encoder, and did BERT style training on it. And now people just take the decoder and do autoregressive training. But what's special about the transformers, which I'm sure most of you are familiar with, is that they have this multi-head attention module where the model learns to pay attention to certain parts of the input in order to build a representation for the next layer. And so these pre-trained transformers, they're usually trained as masked language models, but there's some other tasks we'll do too. Recapitulate biophysical properties. So this is from ESM1B, which is out of Facebook by Alex Reeves and a bunch of other people. So you can show that if you take their input embeddings, and I think this is a t-SNE, down to 2 dimensions, the amino acids cluster by biological property. They also contain a lot of structural information. So here, you take the output from ESM1B, outer concatenate it with itself, and to get an L by L representation, and you can extract structural contacts out of that. And we can also see this with this plot is also for ESM1B. If you take a linear head on top of the representation and train it to predict a class secondary structure, you get a very neat line between the pre-training loss, basically how long you pre-train it for, and how accurate it is on this structural prediction task. So the pre-training loss with the transformer is basically a measurement of how much structural information you've learned. These pre-trained transformers also contain evolutionary information. So meaning the pseudo likelihood correlates with how well tolerated a mutation is at that location. If we mask a location and then take probability of mutant divided by probability of the wild type, that correlates very well with physical measurements. So ESM1B is a Facebook's mass language model transformer. Deep sequence is a VAE that's trained on each protein family separately. And tape is a smaller transformer trained on PFAM. So here are some proteins. Each of these represents an experiment where someone made a bunch of random mutations to this protein and measured the fitness of those mutants in high throughput. And so see higher is better here. And for most proteins, all three of these models have some signal at least. And in some cases, the protein specific VAE deep sequence is better, but in some cases, ESM1B is better. And the Facebook team later trained a model that a variant of ESM with a different training set that's slightly better at these kinds of tasks. You can also use the transformer pseudo likelihood to predict an evolutionary vector field. So here, if I have XA is one protein, XB is another protein, and I look at the likelihood of going from A to B or B to A, that gives me compare those likelihoods, this gives me a directionality. And if I do this for a whole bunch of naturally existing variants, I can make an edge network. Here's one for influenza A nuclear protein. And it turns out that the sinks here tend to be, the sinks of this flow tend to correspond with variants that are the oldest, and they flow towards, in general, towards newer variants. And so flu nuclear protein is a good test case for this because we've been doing flu surveillance for a really long time, and we have many, many variants, and we know exactly when they arose. And this is a virus that evolves fast enough that 100 years or so of data gives you, it's sufficient to see some movement here. We can also use pre-training to guide engineering away from loss of function sequences. So here, this is for green fluorescent protein. So without any pre-training, there's, if we train on only 24 measurements, we basically don't detect, it's prediction. Let me rephrase that. And then we predict the rest of the library. We basically can't use its top predictions to find what are called hits, which are things that are better than the wild type GFP. We train on related sequences. So we take GFP, use MMSeq or HHBlitz to search for related things, and then train a language model on that. You can get some, you get 1.9% hit rate after training on 24 supervised points. If you pre-train on all and then unrelated, you can up that hit rate to 11%. And the reason for that is if we look at PCA of the embeddings after pre-training, but before supervised learning, you can see that the first principal component of the embeddings actually separates out a lot of the non-functional sequences from the functional sequences. And as we fine tune, it just kind of builds off of that initial relationship of learned during pre-training. So this is actually done with an LSTM language model in the Nature Methods paper from last year. Well, the preprint came out long before that. So But most of the current state of the art nowadays uses a transformer architecture with a mass language model. But we also know that out there, just from a scientific perspective, it would be nice to separate the effects of the pre-training task and data set from the neural network architecture use. So in the neural network side, there's things like a multi-layer perceptrons, which are used nowadays in vision and in NLP. There's some interest there. You can use convnets, which are most famously used in vision, but you can also use them on language. There's recurrent neural networks. And then, of course, it's a transformer. And just as an example of very simple tasks that only deal with the sequence, you can look at this mass language modeling task, autoregressive tasks, and then you can do a contrastive task, which is definitely more common with images, but people try to come up with ways to do that for sequences as well. Another reason we might want to consider using something that's not a transformer is that transformers scale quadratically with length in both their runtime and memory requirements. And that's because of that multi-head attention step, which involves matrix multiply between an L by D objects, which is scales as L squared, where L is the sequence length, and that the attention matrix itself is an L by L object. So you need naively L squared memory to store that. So we're going to look at convolutions here, in large part because they scale linearly with sequence length. So this is from a paper about pre-training convolutional models for language and comparing them to transformers. And this is plotting the sequence length on log2 to the number of operations you need in a convnet versus a transformer. So the convolutional architecture we're going to use is very similar to the transformer, except instead of that attention layer, we're going to use what are called bytenet blocks. And bytenet is in this bytenet, a convolution block. If we look at the 1x1 convolution is exactly like the position-wise feed forward in a transformer. So what's really different is the 1x5 dilated convolution, replacing attention. And you have to dilate your convolution to be able to get perceptive field across the whole sequence. And we're going to call this model convolutional autoencoding representations of proteins. And we find that pre-trained CNNs are competitive with transformers on the pre-training task. So, and this is work done with Alex Liu and Nicola Fuzzi at Microsoft Research. So we pre-train UniRef 50, which is the same clustering that ESM1B uses, but we use a different version of UniRef 50 and a different split. And I know that contributes to the general fracturing of these models and evaluations, but there is a reason we had to make our own split, which will be clear later. And just a reminder, the task is we mask corrupt, pass it through a CNN, and reconstruct the original sequence. And so here we test the CARP architecture and ESM's transformer architecture across several orders of magnitude for parameters on our data set, and those are the orange and blue lines. So you can see across several orders of magnitude, the architecture doesn't seem to affect the pre-training result that much. And then we also trained CARP 640 million, which is that bottom right point in blue, which has about 640 million parameters, to compare with ESM1B, which has about 650 million parameters. So those losses are close, but they're not directly comparable because they're on different test sets with different train sets. So the only conclusion here is that things seem to be close. So here's CARP again. And just like a transformer, pre-trained CNN can learn structure. So here we're going to compare directly to ESM1B. So first task is long-range contact prediction. And here's these results for Cameo. And we're pretty close there. We also do remote homology. So ESM1B is a bit better here, but we do definitely learn something here. So remote homology is given the protein sequence, classify which calf I think superfamily it's in. And then this is three-class secondary structure, predicting whether each residue is part of a beta sheet, alpha helix, or a loop. So they're pretty much identical results. And just like ESM1B, the longer you pre-train, the more structural information you encode. So here I'm showing 3 different model sizes for CARP with 640 million parameters, 66 million parameters, and 600,000 parameters. And then if it's a solid line, that means I'm fine-tuning the model all the way through. And the dashed line means I'm freezing the pre-trained model and only learning the weights for the model on top that predicts secondary structure. So here, the horizontal lines are without pre-training. If I just fine-tune or train those weights on top to predict secondary structure, it's interesting here that fine-tune all the way through helps, which makes sense because you have more parameters to use, but also the more parameters you have, the better you do, even without pre-training with a ConvNet. And as we pre-train, the pre-training loss corresponds very well with the final accuracy. So just like in a transformer, when you pre-train a CNN with a mass language modeling task and sequences, you are encoding structural information. CARP is also a zero-shot mutation effects predictor. So a quick reminder what this means. People do what are called deep mutational scans, where you make a few changes to your protein sequence and measure the effect on its fitness. And without retraining the model, we mask every location where there's a mutation and then compare the likelihood of mutation to the wild type. And we're going to evaluate on deep sequence, which is a set of 41 deep mutational scan data sets that were helpfully collated by John Graham and the Marks lab. So average across these, we get a Spearman correlation of 0.49. ESM1B, which is trained on UniRef 90s and UniRef 50, and is an ensemble of 5 models, is slightly better. ESM1B is slightly worse. And if we just use a position-specific score matrix, which is basically a site-independent model based on an MSA, that's also worse. And we beat ESM1B on 18 out of the 41 data sets, so a little less than half, a little bit more than half on ESM1B, and much more than half against the site-independent model. We also find that zero-shot performance usually improves with more pre-training. So here again, those same 3 model sizes. Here we don't fine-tune anything. This is zero-shot prediction. C and UniProt refer to 2 variants of what we use as an input sequence. And so on the X, I'm plotting the pre-training loss, and the Y is a zero-shot Spearman row for the E. coli beta-lactamase data set from Osmeyer 2014. And for most of the data sets in deep sequence, we get something that looks like this, where the bigger model is better, and the better you do on pre-training, the better you do on zero-shot. However, there are a few exceptions. Some look like this, where we improve for a while, but then more pre-training makes things worse. And then some are like this, where almost immediately you make things worse with more pre-training. So we are investigating why this happens and what might be behind it. But this is an area of ongoing research. And these are plots I have not shown before in the talk, and they're not in the paper. So bonus information for you all. And finally, for protein engineering, what we really care about is what I call out of the domain generalization. Some examples of this might be, I train the model on single mutants and predict on double, triple, quadruple mutants, etc. Or from the single double mutants, I want to learn higher order things, or single, double, triple, and test in higher order things. So this is an easy one where you just take this as split right 80-20. You don't really see that come up as often in these protein engineering campaigns. Another one that's of interest, of possible interest, is train in everything that's not as good and predict things that are better, because that comes up. For a lot of protein engineering cases, what you really care about is, can you find improved variants? And some reasons why we care about things like this. One is, in most cases, we can measure many or all of the single mutants to a protein of interest. So if your model, for example, needs 80% of the single mutants to learn a good model for the other 20%, that's just not very useful, because I'm just going to measure all of them. This doesn't scale though with bigger libraries. For example, if I've picked 10 sites to mutate, the theoretical library is 20 to the 10th, which is a very big number. And if I randomly sample from that library, almost everything I sample is going to be non-functional. So being able to, so I can't naively sample from that library, but if I can sample from a library of, say, 1 or 2 mutants where many things will be functional, and extrapolate to this bigger space, that's very useful. I also think from just a biological and machine learning perspective, this is a useful thing, an interesting thing to be able to do. We do find that for CNNs, as for transformers, pre-training does improve this kind of generalization in many cases. So here, in a specific case, we have a protein called GB1. It's highly epistatic. It's mutated at 4 positions. We have most of the 160,000 theoretical library measured from a paper from, I think, 2016 by Wu et al. And the paper shows more evaluations, but here I'm just going to show 1 versus rest and 2 versus rest. So we're doing Spearman with pre-training. So 1 versus rest, objectively, a Spearman of 0.19 is really bad. With 2 versus rest, you do much better. ESM1B does better in 1 versus rest, but much worse in 2. And without pre-training, those models do worse. And in fact, without pre-training, they do worse than a tiny CNN, probably because they way overfit. But with pre-training, they beat the tiny CNN baseline. So both architectures benefit from pre-training. Which one is better depends on the task. And I don't show this in this table, but we do find that CARP does better if you fine-tune all the way through, as opposed to freezing the pre-trained weights, which is a different conclusion than what's been published elsewhere for other models. Oddly enough, even though we find that pre-training helps, out-of-domain performance mostly does not improve for pre-training. So this is a different task. This is a thermostability task. Once again, we have those same 3 models, fine-tune or freeze the weights. And you can see that the relationship between the pre-trained loss and how good the downstream performance is kind of not a straight line either direction. This is also an area of investigation. This is also not in the paper, so more bonus content. I'll stop here for questions before I move to the second paper. Philip?
Hi, Kevin. Yeah, thanks so much for the interesting talk. So I was wondering, I might have missed this, but do you guys experiment with the percentage of the sequence that you mask out and the length of the masked out tokens?
No, so we just use the same BERT task that ESM1B uses, which is you mask 10% of the tokens, you mutate 2.5%. And there's another 2.5% that you keep the same, but you evaluate there just so it doesn't always learn to change everything.
Mm-hmm.
Which is the same as a BERT task.
Yeah, it was just interesting. I wonder if there is like a, when you go from one domain to another, like in the masked autoencoder paper from Kaimin Key, when they applied, I guess, this masking strategy for images, they found that like if you mask out 75% of the image, it's a lot better. So that's something I was thinking along the lines.
Yeah, 75% is probably a little high here, but there's definitely some creativity you can do around like not choosing masking completely randomly, masking out bigger chunks, right? Or there's another paper out there where we know that these masked language models for proteins learn a lot of these pairwise relations, but they actually explicitly ask it to also to recapitulate the pairwise relations in the loss, where they, in addition to predicting logits over the 20 amino acids, they take some pairs of positions and ask to predict the 400 possible pairings there and assign probabilities to those. And they get some interesting results out of that. So there's a lot of creativity in these kinds of models that I haven't explored here.
Right, yeah. It seems like there's a lot of possible things to iterate on in this space.
Yeah.
Thanks.
I also wanted to ask a question. So when you say you're predicting without pre-training, what does that mean? What are you training on?
Just the data from the downstream task. It's a supervised task.
And the task would be to predict the fitness of the mutants. Is that the task in the last slides that you were talking about?
Yeah.
Got it. Thank you.
So this is not a zero-shot task.
I'm just assuming that like the amount of data there is much less than the pre-training on the protein data, right? So you take kind of those really big models that can kind of pre-train a whole protein, and you give it like very small amount of data.
So if you have 4 sites, the number of possible single mutants is 80, right? So that's which is a much smaller number than 42 million.
Yep. Thank you.
And do you mind commenting a little bit more on this OOD task? I might have missed this, but what is the actual out of distribution set?
So this one is not as out of distribution, actually. We have measurements for the Melton task. We have measurements for thermostability for a bunch of different proteins. We've clustered them so that you don't see similar proteins during training. So that's the only way it's out of distribution. And you're just predicting the thermostability from the sequence.
Oh, I see. So it's okay. So it's like
it's not as aggressively out of domain
split.
as it's not as but it's not as aggressively out of domain as the GB1 task I showed in the previous slide.
Right.
But the GB1 ones also look like this. I just cherry picked one that was representative.
Thanks.
So hold on. Then for the without pre-training, you train from the data from the same task, right?
Yes. Yes.
So in that case, where does the out of domain coming from? Is that you're testing on very different proteins that were in a training set?
Yes. So like for the Melton task I'm showing here, we've clustered by sequences, by sequence similarity. So you're not seeing anything with more than like 50% similarity during training. For the GB1 tasks, you're training on, say, single mutants and predicting double, triple, quadruple mutants.
Gotcha. Yeah. That, and it makes sense that kind of pre-training and seeing like a large variety of proteins actually helps the model to predict this even on the like this property even on the proteins it has not seen the terminal stability values before.
Yep. Cool. So we'll go on and we're going to add in some structural information. So this project is with Hugh Ye, who's a PhD student now at University of Chicago, MD PhD student. ambitious. And Niccolo Zanichelli, whom you all know well. And so, reminder of CARP, we only deal with sequence. We have 42 million sequences from URF50. Turns out that, so there's this task on mask inverse folding, and this is inspired by work from John Graham's 2019 NERDS paper is really the first one to do this, where you take a neural net, you give it the structure, and then from the structure, you generate the sequence. And they do it autoregressively. And recent work calls this inverse folding, much to Sergey's chagrin. But we call this mask inverse folding because we actually do a BERT task instead of an autoregressive task. So we train here on 19,000 structures and sequences from CAF. We're going to encode the backbone, so you can't give it the side chains because that would give away the amino acid identities. And we're going to encode the C beta distances as well as the bond angles. So distances, angles, along with the mask sequence into a graph neural network, and then use that to recover the original sequence. So here's CARP, and we call this mask inverse folding. And on the CAF test set, so perplexity lower is better, with only 19,000 examples and 3.4 million parameters. If you get structure, you get a lot of information, your perplexity is 4.95 versus 7.06 for the big pre-trained CNN. And this is why we had to make up our own training test split for the CNN, because we held out the test set from CAF from our training set for the CNN to make this a fair comparison. Okay, and then, but there's these 42 million sequences that are being left out. Can we somehow incorporate them to make this even better? So we call this mask inverse folding with sequence transfer. So we only train on CAF again, but we use that pre-trained CARP 640 million parameter model to embed the mask sequence and then output its outputs to the GNN. And we freeze the CARP component for computational convenience. And so with only 3.4 million trainable parameters, you can drop that perplexity down to 4.08. And this model is also a strong zero-shot fitness predictor. So that we've seen before. So here are models. We have the CARP model, and then we have ESM1V, which is an ensemble of 5 transformers. If we add in structure, we have mask inverse folding, and then we add in sequence transfer as well. We have MIFST. So here's the same deep sequence Spearman correlations. So mask inverse folding by itself actually doesn't do great here, probably because the CAF dataset is so deduplicated against by sequence homology that it's hard for it to differentiate between close relatives. But if you add in sequence transfer, you do better than CARP or MIF by itself. We also evaluate on a data set of small synthetic proteins. We call this a Rocklin data set because it was originally designed and measured by Gabriel Rocklin. And here, sequence only doesn't do that great. If we add in structure or structure and sequence transfer, we do a lot better. So that last model that just showed up out of nowhere is a model that came out right before we preprinted ours. It's a very similar idea. They have two changes. The one is they use a GNN architecture called GVP, or Geometric Vector Perceptron, which is a newer architecture than the one we used and should do better. But the bigger change is that in addition to training on structures from CAF, they also use AlphaFold to predict another 12 million sequences, and they augmented their training with that, which helped a lot too. And this paper came out by Chloe Xu. They call this ESMIF, ESM inverse folding. It's a great paper. You should go read it. We also evaluate on a deep mutational scan of the receptor binding domain for COVID bound to ACE2. Here, the sequence only models do really poorly, and augmentation seems to really help here too. So sequence conditioning helps sometimes, and at least for this complex here and some other complexes too, the augmentation seems to help a lot. They also use a different masking scheme. They don't mask. It's mostly autoregressive, which makes it better able to deal with variable length things. One interesting thing is these zero-shot predictions are often better using AlphaFold 2 structures than the PDB structures. So here comparing the two models and then the data sets using PDB or AlphaFold 2. So deep sequence, on deep sequence for both MIF and MIF-ST, we do a lot better using AlphaFold 2 structures than using the native PDB structures for each system. It's unclear to me why this is. We also do this for GP1. It's that same 160,000 things, 160,000 mutant theoretical library. Objectively, these are all pretty bad, but we do get better correlations using the AlphaFold 2 structure. And here that picture just shows the AlphaFold 2 structure and the PDB structure are very, very similar for this protein. For receptor binding domain, using the AlphaFold 2 structure hurts a lot. But then I looked at the structure I have from AlphaFold and it puts the RBD in the wrong place. So that explains why that might happen. So if you have a good AlphaFold 2 structure, it seems to do really well for zero-shot prediction. And depending on the task, using MIF with sequence transfer can improve even more on that out-of-domain generalization. So here's that same GB1 task. We're looking at the same two splits. So CARP with pretraining, this you've seen before. Here's MIF. So MIF actually doesn't do better on 1 versus rest or 2 versus rest, but if you add in the sequence transfer, it does better on both. And without pre-training, MIF is very, very, very bad actually. And the MIF-ST architecture is very unstable without pre-training. It predicts the same output for every out-of-domain thing. And here's that small CNN baseline again. So structure and sequence transfer helps a little bit here. I said the naive MIF-ST predicts all the same values, which is why you get nons. Also came up with a new task, which is we have an enzyme when to predict neoantial selectivity given the silicon containing substrate. And this library contains mutations at 7 positions, but we're going to take predictions that train on measurements from predictions those 4 sites in green and predict for variants that have mutations at all 7 positions. And we call this the RMA nod data set after the enzyme. So here, you need both for both CARP and MIF. Pre-training doesn't actually seem to help much at all. And if anything, it makes it a little bit worse. But if you have MIF-ST, the pre-training does seem to help a lot. And structure and sequence and structure pre-training are both necessary here. You can try these models. If you get bugs in these, you should submit an issue or ping me and I can usually fix it very quickly. But you can try all of these models on GitHub. The weights are in Zenodo. The code's on GitHub. And a little bit of outlook here. So kind of big picture is we want to use multiple data modalities with some proteins. So during initial model training, we take natural protein data and train some model. in that model, the data might include sequence, as in CARP, also annotations, things like Go annotations or the species or localization tags. The structure as in MIF. We might get some chemical information or substrate information. There might be free text. You're kind of limited by your imagination here. And in my head, there might be other ways, but there's kind of two ways you can use these pre-trained models. One is as pre-trained protein fitness predictors. So you fine tune it on a small set of measurements and go find things that do better. Or the cooler thing is you specify the function you want and you generate some sequences that you can then go make in the lab that will do this without needing a starting point. Then some acknowledgments. Most of this work was done as part of biomedical machine learning group at MSR New England. Because we all started in the pandemic, there's not a picture of all of us, so you just get a logo. We also run a machine learning for protein engineering talk series. I recognize that 2 of these talks are in the past, but the next one is July 5th. And then there's a moderated panel with those 3 speakers. And I'm also pretty excited for the next set of talks after that, which we'll announce soon. And now we will I'll take more questions.
There's a question in the chat.
Is that a question or just?
A little up there, Sergey is asking if AlphaFold 2 dataset includes PDB, which I guess could explain the effect that you're seeing, that it helps.
I'm not sure what AlphaFold 2 dataset Sergey means here.
Oh, I guess I could ask. It was that slide you were wondering about. Why did it improve when you used AF2 versus PDB dataset?
Oh, not dataset. Those are actual structures. Because the graph neuron then uses an input structure, right? So you can use the experimental structure
to.
Yeah. Or I just ran them through, or I can run the sequence through Colab Fold. Which is a very nice tool.
So yeah, I wanted to ask about that as well. So does it seem to help in the case where the sequence, like the real structure and AlphaFold structure are close to each other, or does it seem to help when they're different?
It does seem to help even when the structures are very close to each other.
Wow.
This is also something to investigate further. Justin asked if I've experimented with not freezing the carboids in the FSD. I briefly experimented with that. 1, it takes a lot longer to train, but 2, it overfits really quickly. So it does really well on the training set. But it overfits in like 1 epoch.
Thank you, Kevin, so much for the talk. If I can ask one thing, is whether you think it is worth working on other protein language models still, apart from ESM and CARP. Like, is the limit high enough that we can improve upon them by a fair amount, or are we approaching the limit imposed by the coevolutionary information present in those pre-training data sets, and then maybe it's best to work with more supervised learning approaches?
Yeah, so my take is, given, so this language modeling task in general seems really good at this, right? This is a nice trend, but it's not And even on the zero shot things is a pretty nice trend. But I think it's pretty clear that on these out of domain things, the trend is not nearly as good. And that seems to be the case no matter which architecture you use, which tells me we should be looking at different tasks that will generalize better for these kind of engineering tasks. That's my opinion, at least. But maybe if someone trains a billion parameter one, will prove me wrong. Who knows?
Awesome, thank you. I guess there are no other questions. All right, and I will thank you, Kevin, a lot for the presentation, for the community itself, and yeah, I think.
Thank you all for coming.